Imagine a world where we can edit our DNA to cure diseases, grow better crops, or even design the future of humanity. The power to edit DNA is here, and it's shaping our future today. CRISPR technology is changing everything we know about genetics. This incredible tool gives scientists the power to edit DNA with unmatched precision, opening doors to groundbreaking treatments for genetic disorders, stronger crops, and even new ways to fight diseases. But with great power comes great responsibility. While CRISPR holds amazing potential, it also raises big questions about ethics and how far we should go. Welcome to the nautical depths, and in this video we'll explore how CRISPR works, its exciting possibilities, and the challenges it brings to our future. Origins and history. In 1993, Dr. Francisco Mojica from the University of Alicante in Spain identified unique repeating DNA sequences in bacteria. He named these regions CRISPR and suggested they were part of the bacterial immune system. This theory was later proven in 2007 by a team led by Dr. Felipe Horvath, who showed how this system helps bacteria defend against viruses. Now it has become famous as a precise gene editing tool, but it took years of research to understand and develop its full potential. Dr. Jennifer Doudna from UC Berkeley and Dr. Emmanuel Charpentier from the Max Planck unit in Berlin made the groundbreaking discovery in 2012. They published a research paper explaining how the CRISPR-Cas9 system from bacteria could be used for editing genes. This discovery was so significant that they won the Nobel Prize in Chemistry in 2020, making history as the first all-female team to win the prize. Other scientists also contributed to advancing this technology. Dr. Feng Zhang at the Broad Institute of MIT and Harvard worked on using this technology in complex organisms and discovered new Cas proteins. Dr. George Church at Harvard Medical School demonstrated CRISPR's use in human cells. Biochemist Virginijus Siksnis from Vilnius University in Lithuania independently showed that CRISPR could edit genes in various organisms. Rapid progress in CRISPR applications. Since Doudna and Charpentier's work in 2012, this technology has evolved rapidly. It has been adapted for various uses, like editing RNA, studying genes in live cells, and developing diagnostics. CRISPR has been successfully used in humans and other organisms for treating genetic conditions. In 2019, the first clinical trials using this technology started. Scientists edited cells from patients with sickle cell disease and reintroduced them into the body, a method known as cell therapy. In 2020, CRISPR took another leap forward with gene therapy. This involved directly injecting CRISPR tools into patients to treat hereditary blindness. Now, you know the history and origins of this revolutionary breakthrough, but how does it work in the first place? Well, let us make it simpler for you. How it works: CRISPR, short for clustered regularly interspaced short palindromic repeats, is part of a bacterial immune system that helps bacteria fight viruses. It is like a natural immune system for bacteria and archaea. It helps them fight off viruses using a special enzyme called Cas9, which works like tiny molecular scissors. When a virus attacks a bacterium, the bacterium uses Cas9 to cut out a piece of the virus's DNA, called a protospacer. This piece is stored in the bacterium's DNA as a memory of the attack. Over time, the bacterium builds a collection of DNA fragments from different viruses it has encountered, like an immune record. These fragments are kept between repeated sequences, giving CRISPR its name. If the same virus attacks again, the bacterium can quickly recognize it and destroy it. Cas9 is guided by a molecule called guide RNA gRNA. This gRNA is made of two parts. One part matches the virus's DNA, the key rRNA, and the other acts as a scaffold to hold everything together, the tracer RNA. Think of Cas9 as scissors and the gRNA as the instructions telling Cas9 where to cut. Before cutting, Cas9 searches the virus's DNA for a specific short sequence called PAM. Once it finds PAM, Cas9 checks the DNA nearby to see if it matches the target. If it matches, Cas9 makes a cut in the viral DNA, breaking it apart. Since viruses can't repair their DNA, this cut stops them from functioning, protecting the bacterium. Scientists like Dr. Dodner and Dr. Emmanuel realized they could use this natural system for gene editing. By giving Cas9 a custom guide RNA, they could direct it to cut DNA at any desired spot in the genome of any organism. This discovery changed everything. To make the system simpler, researchers combined the two parts of the guide RNA into one molecule called single guide RNA. This innovation made it easier and cheaper to use CRISPR for genetic editing. Scientists can now program Cas9 to target specific DNA sequences as long as the PAM sequence is present. When CRISPR makes a cut in the DNA, the cell tries to repair the damage. It uses a template to fix the DNA precisely. Scientists can use this to insert new genes or fix faulty ones. By combining CRISPR's precision with this mechanism, scientists have created a powerful tool for editing genes, opening up endless possibilities in medicine, agriculture, and beyond. Transforming medicine with cell and gene therapies. CRISPR technology is revolutionizing medicine with its potential to treat and even cure genetic diseases. Conditions like neurodegenerative diseases, blood disorders, cancer, and vision problems are now within the scope of advanced treatments. For instance, the first CRISPR-based cell therapy trial was conducted in 2019 to treat sickle cell disease. This therapy restored fetal hemoglobin levels in patients, eliminating the need for a functioning adult hemoglobin gene. In 2021, another breakthrough came with a CRISPR trial for transthyretin amyloidosis, a neurodegenerative condition, showing highly promising outcomes. This technology is also making waves in pediatric cancer treatments, enabling advancements like creating chimeric antigen receptor CAR T cells. These engineered immune cells are programmed to attack cancer more effectively. The future holds even greater promise. Clinical trials are exploring treatments for thousands of genetic conditions such as Huntington's disease, Tay-Sachs, cystic fibrosis, and even early-onset Alzheimer's. As research continues, CRISPR is paving the way for transformative therapies that could change countless lives. CRISPR is a diagnostic powerhouse. CRISPR isn't just a tool for gene editing, it's also a game changer in disease diagnostics. During the COVID-19 pandemic, it played a crucial role in both potential treatments and virus detection. Another exciting development is Mammoth Bioscience's Detect-TR system, which uses this system to find and identify viral genetic material. These diagnostics rely on CRISPR's ability to locate specific genetic sequences, making it highly effective for identifying diseases. Beyond infectious diseases, CRISPR-based methods are being developed to detect genetic conditions. For instance, Dr. Kiana Aaron's research combined CRISPR with cutting-edge technologies like graphene and transistors to create a chip capable of identifying genetic mutations linked to human diseases. These advancements highlight CRISPR's incredible versatility in the medical field. Revolutionizing agriculture with CRISPR. CRISPR is set to transform agriculture by creating crops that are better suited to modern challenges. Scientists are using this technology to develop plants that resist diseases and survive extreme weather. For example, researchers are collaborating with Mars, Inc. to create cacao plants that can withstand diseases. It is also being used to extend the shelf life of fruits and vegetables, helping reduce food waste. Imagine fresh produce that stays edible longer while remaining affordable. Thanks to this technology, this could soon become a reality. With such innovations, this technology could provide healthier food options, reduce farming challenges, and ensure a more sustainable future for agriculture. Boosting bioenergy with CRISPR. Bioenergy, a promising alternative to fossil fuels, is getting a boost from this technology. Producing biofuels on a large scale has been challenging, but gene editing is helping overcome these obstacles. Scientists have used CRISPR to enhance lipid production in algae, significantly increasing its potential for biodiesel creation. Similarly, yeast strains are being edited to tolerate harsh production conditions, improving ethanol production efficiency. These breakthroughs show how CRISPR is helping us create cleaner, sustainable energy sources. By improving biofuel production, it is contributing to the fight against climate change and supporting the transition to greener energy solutions. Moral dilemmas of gene editing. Although this technology holds incredible potential, but it also brings up serious ethical concerns. One major issue is the possibility of editing human embryos, which could lead to designer babies with traits like eye color or intelligence being chosen. This raises questions about fairness and the potential for inequality. Another concern is the long-term effects of gene editing, which we don't fully understand yet. It's important to consider how far we should go with CRISPR and make sure that its use is responsible and benefits everyone without causing harm. In a nutshell, CRISPR technology has opened up new possibilities, offering hope to improve our world. However, it's crucial that we balance innovation with caution, protecting the future of humanity. So, what are your thoughts on this technology? Tell us in the comments below, and meanwhile, subscribe to the Nautical Depths for more infotainment videos like this one.